An animal is living in the ocean right now that evolved a mind completely separate from ours. Same planet, zero shared ancestors for over 500 million years. It has 3 hearts, blue blood, 9 brains. It is colorblind, yet it disappears into its surroundings in under a second. It can impersonate a dozen different animals on the fly. It escapes from supposedly locked tanks, raids its neighbors for food, and returns before morning. Scientists have sequenced its genome and found it has more genes than a human. If you enjoy this, like and subscribe. We cover things like this every week. Today, we go inside the most alien mind on Earth. Prepare yourselves. We begin. 500 million years ago, something happened twice. Intelligence evolved, and the second time, it looked nothing like the first. Every smart animal you have ever heard of shares a family tree with you. Dolphins, crows, elephants, chimpanzees, they all inherited their brains from the same ancient ancestor. The same basic blueprint, refined over millions of years, one path to a mind. Then there is the octopus. The last creature humans and octopuses share as a common ancestor was a flat, eyeless worm crawling through shallow prehistoric mud. That was before plants had colonized land, before fish existed, before almost anything we would recognize as an animal had appeared on Earth. And from that point, the two lines split completely. Vertebrates went one direction, cephalopods went another. For 500 million years, those two lines never shared a trick, never traded a gene, never borrowed a single piece of brain architecture from each other. And yet, at the end of that enormous stretch of time, both lines produced something capable of solving puzzles, remembering faces, using tools, and making decisions in real time. That is the most important thing to understand about the octopus before we go any further. It is not a primitive creature that stumbled into a few clever habits. It is the product of a completely independent experiment in building a mind. Evolution ran the intelligence test twice, in total isolation, and got 2 totally different answers that both work. Most of the animals we study to understand intelligence share so much with us that their similarities are almost expected. A chimpanzee brain looks like a smaller, simpler version of a human brain. A crow brain follows the same general layout. Compare those to an octopus, and you are looking at a different universe. The wiring, the structure, the relationship between body and brain, all of it was solved from scratch. Here is where that gets strange. When scientists study how intelligence works, they use the family resemblance to fill in gaps. They assume that if a dolphin learns something in a way similar to how a human learns it, the underlying mechanism is probably similar too. That assumption breaks completely when you cross to the octopus. There is no family resemblance to lean on. Every discovery has to be earned from zero, and the discoveries keep coming. In the last 2 decades, research on octopus cognition has accelerated faster than almost any other field in animal behavior science. Every year, octopuses do something that forces scientists to rewrite the rules about what intelligence requires, what consciousness might mean, and what kinds of minds are even possible. A team at the University of Chicago sequenced the first complete octopus genome and described it as the first genome from an alien. That word choice was not casual. The lead scientists meant it as precisely as scientists ever mean anything. The genome of an octopus is so unlike any other animal genome that its closest comparison is something from a different world entirely. The octopus has 33,000 genes. Humans have around 20,000. The extra thousands appear in places no other animal has them, doing things no other animal does. And it all fits inside a body with no bones, no rigid frame, no fixed shape, and no permanent color. The questions that raises are not simple ones. If a mind this sophisticated can grow in a body this different, what exactly does intelligence require? What is a brain? Really? What is a thought? The octopus does not answer those questions. It just makes them impossible to ignore. And the body this mind lives in is even stranger than the mind itself. The octopus body should not exist. Run through the specifications, and it reads like something invented for a science fiction film. No skeleton, no bones, no cartilage, no rigid structure of any kind. The entire body is soft tissue, capable of squeezing through any opening larger than its beak, the only hard part it has. An octopus the size of a basketball can push itself through a hole the diameter of a quarter. Now add the circulatory system. 3 hearts. 2 of them, called branchial hearts, sit near the gills and pump blood forward to collect oxygen. The third, the systemic heart, collects that freshly oxygenated blood and drives it through the rest of the body. Here is the part that sounds like an engineering mistake. When the octopus swims, the systemic heart stops beating completely. The effort of jet propulsion shuts down the main pump. This is why octopuses almost never swim long distances. Swimming for them is not graceful gliding. It is a brief emergency sprint that physically exhausts their cardiovascular system. Crawling is their natural mode. Slow, controlled, arm over arm along the seafloor. The blood these 3 hearts push around is blue. Not metaphorically, not slightly bluish. Blue. Human blood is red because it uses a protein called hemoglobin to carry oxygen. Hemoglobin is built around iron, and iron turns red when it binds to oxygen. Octopus blood uses a completely different protein called hemocyanin, and hemocyanin is built around copper. Copper binds oxygen and turns blue-green. The ocean floor where octopuses live is cold and low in oxygen. Hemocyanin performs better than hemoglobin in exactly those conditions, delivering oxygen more efficiently when the temperature drops and the supply gets thin. 3 hearts and blue blood are already enough to make the octopus sound like a creature from another planet. But the body goes further. The skin is not passive. It is not just a covering. The octopus skin is an active, controlled system with roughly 230 tiny color cells packed into every square inch. Underneath those sit reflective cells that bounce light in specific directions. Beneath those sit white scatter cells that act as a backdrop. The whole system works like a 3-layer screen, and the octopus can update every pixel in real time. The texture changes too. Tiny muscular bumps, called papillae, can be raised or flattened in under a second, turning smooth skin into something that looks like gravel, coral, or rock. The octopus does not just change its color to blend in. It reshapes its surface to match the physical texture of whatever it is sitting on. The arms themselves are not simple appendages. Each one contains hundreds of suckers. Each sucker can grip, taste, and sense touch independently. The arm does not just hold things. It reads them chemistry, texture, temperature, and sends that information toward the brain in real time. And none of this body has a fixed shape. The octopus has no up or down, no front or back that it has to maintain. It can flatten into a pancake, stretch into a tube, ball itself up, or flow around a corner like liquid. A body this flexible, built around this much soft, living tissue, operating without a single rigid support, should be fragile. Instead, it is one of the most durable and adaptable body plans in the history of animal life. It has been running, largely unchanged, for hundreds of millions of years. What powers it goes even deeper than the body itself. The octopus has 9 brains. That sentence requires a little unpacking, because the 9 brains are not 9 identical copies doing the same job. They are a network, 1 central brain and 8 smaller ones, 1 for each arm, and together they run a system of control that no engineer has ever replicated. The central brain sits between the octopus's eyes. It handles the big decisions: hunting strategy, threat response, social interaction, memory. It processes what the eyes see and coordinates overall behavior. By itself, it would already be remarkable. An octopus central brain has more neurons than a mouse brain, packed into a structure smaller than a walnut. But 2/3 of all octopus neurons are not in the central brain. They are in the arms. Each arm contains its own dense nerve cord running the full length from base to tip. That cord has enough local processing power to receive information from the suckers, decide how to respond, and carry out a physical action, all without sending a signal up to the central brain first. The arm can taste something, judge whether it is worth grabbing, and close its suckers around it before the central brain has even been notified. This is not a minor delegation of tasks. It is a fundamentally different architecture of intelligence. In a human body, the brain is the sole decision maker. Your fingers do not decide to pick something up. Your brain decides, sends a signal down the spinal cord, and the fingers execute the command. The chain of authority is strict and runs from the top down. Every action begins centrally. An octopus runs the opposite model. The arms are not waiting for orders. They are making their own local calls, reporting back when it seems relevant. The central brain sets the general direction. The arms handle the details. This means an octopus can do 8 different physical things at the same time, each driven by a different arm brain, without any of those 8 things creating a traffic jam in the central brain. Imagine 8 separate versions of your hands, each capable of problem solving on its own, all reporting back to a coordinator who only steps in when the overall situation requires it. Scientists studying this system found something even stranger. When an octopus arm is removed, either cut off in a lab setting or lost in the wild to a predator, the severed arm keeps moving for up to an hour. It keeps reacting to touch. It keeps trying to pass food toward where the octopus's mouth used to be. The arm does not know the body is gone. The local brain in the arm keeps running its program independently. That is not a reflex in the usual sense. A reflex is a single, fixed response to a single trigger. What the severed arm does is a sequence of goal-directed behaviors, sustained over time, driven by local neural processing. For robotics engineers, this architecture is a dream. A robot arm that can navigate tight spaces, adjust its grip, and handle unexpected obstacles, all without sending every micro-decision back to a central processor, would solve some of the hardest problems in machine design. The octopus solved that problem 500 million years ago. What the arm brains do, moment to moment, is remarkable. What they do while the octopus changes color is stranger still. Put an octopus arm in a dark box and touch it with something rough. The arm will move. It will explore the object. It will pass information about that object upward through the nerve cord. And crucially, none of that requires the central brain to do anything at all. The arm handles the exploration independently, like a scout gathering data in the field. This is not unique to a specific species or a specific behavior. It is the baseline design. Every octopus arm operates with a degree of independence that has no equivalent in the vertebrate world. Each of the suckers lining the arms and a large octopus can have more than 2,000 of them contains chemoreceptors. These are chemical sensors, similar in function to taste buds. When a sucker presses against a surface, it does not just grip, it reads. It detects what the surface is made of, specific molecules, traces of prey, chemical signatures left by other animals. The sucker reports this to the arm's local nerve cord. The arm processes the combined chemical read from all its suckers simultaneously. It builds a picture of the environment through chemistry, not just touch. This is how an octopus hunts in the dark. It does not need to see what is inside a crevice to know whether something worth eating is in there. The suckers reaching inside gather enough chemical information to make that call without any light at all. The arms also control sucker spacing and force with remarkable precision. A researcher once watched an octopus unscrew a jar lid in a laboratory tank. The animal had never encountered a jar before. It had no training. It pressed its suckers against the lid, felt the resistance, adjusted the angle of force, turned the lid in the correct direction, and opened the container in under 2 minutes. Each sucker independently calibrated its grip. The arm coordinated the whole rotation without requiring the octopus to plan the motion in advance. What made this particularly striking was what happened next. When the same octopus was presented with the same jar the following day, it opened it in under 30 seconds. It had remembered, and the memory was precise enough to cut its working time by more than 3/4. The arm itself is thought to hold some of that motor memory locally. Scientists believe that certain repetitive physical tasks, unscrewing, reaching, gripping, become partially embedded in the arm's own nerve cord over time, the way muscle memory works in humans. The difference is that octopus muscle memory may be literally stored in the arm, not just in the central brain. There is a consequence to this distributed architecture that scientists are still working to understand. When an octopus makes a decision, where exactly does the decision come from? If the central brain says reach for food, and the arm decides how to do it, and the suckers decide what to grip, and the local nerve cord coordinates the whole sequence who decided, the honest answer is that all of them did, together, in parallel, without a single moment where authority passed from one to another. That model of mind, spread across a body rather than concentrated in one location, is something humans have never built, never evolved, and barely understand, even in theory. The arms are doing all of that, and at the same time, the skin wrapped around them is doing something that should be physically impossible. The octopus is completely colorblind. One type of photoreceptor, one visual pigment. Scientists have confirmed this repeatedly across dozens of species. By every standard measure of color vision, the octopus sees the world the way a black and white photograph looks. It detects brightness and contrast. It detects shapes and edges. It does not see the difference between red and green, blue and orange, or any other pair of colors. And yet, watch an octopus settle onto a patch of coral and your jaw will drop. Within 1 second, the animal vanishes. The skin shifts through a sequence of colors, browns, reds, oranges, creams that match the surrounding coral with photographic accuracy. The texture of the skin rises in the exact pattern of the nearby rock surface. The animal becomes invisible, not just from one angle, but from every angle, to predators with full color vision. This is one of the strangest mysteries in all of biology. A colorblind animal matching colors it cannot see, faster than the eye can track, more accurately than any human artist could paint. The question of how has driven scientists to design increasingly clever experiments, and every answer they find opens into a new, deeper question. What researchers know for certain is that the octopus skin contains roughly 230 tiny color cells called chromatophores packed into every square inch. Each chromatophore is a sac of pigment attached to a ring of tiny muscles. When those muscles tighten, the sac stretches wide and its color becomes visible. When the muscles relax, the sac contracts into a dot too small to see. The octopus controls these individually, updating the pattern across its entire skin, the way a screen updates pixels. Underneath the chromatophores sit reflective cells called iridophores. These do not contain pigment. They contain stacked layers of thin plates that reflect light at specific angles, producing the blues and greens that the pigment sacs alone cannot make. Beneath those sit white scatter cells that create the pale background that all the other layers work against. 3 layers, each doing a different job, all updating simultaneously. The octopus controls all of this, actively, precisely, in real time. And the control system runs from a brain that receives no color information from the eyes. So what is the brain telling the skin to do, if it cannot see what the skin should look like? That question does not have a clean answer yet, and the leading candidates are stranger than the mystery itself. Something in the way the octopus processes visual information, or something in the skin itself, is solving the color problem through a route the brain alone cannot explain. Something outside the central brain is seeing color, and the evidence for what that something might be is exactly what keeps marine biologists awake at night. In 2015, a research team made a discovery that the biology textbooks had not prepared anyone for. Octopus skin contains the exact same light-sensing proteins found in eyes. These proteins are called opsins. In the eye, they sit inside photoreceptor cells and react to incoming light by triggering a nerve signal. That signal travels to the brain and gets interpreted as vision. Opsins are what make seeing possible. They were not supposed to be anywhere else. The team, publishing in the Journal of Experimental Biology, confirmed that octopus skin contains functional opsin proteins distributed across its surface, not just in one patch, across the entire skin. And when they isolated strips of octopus skin completely removed from the animal, with no connection to any nervous tissue, and shone light on them, the skin reacted. The chromatophores expanded. The skin changed its pattern in response to light, with no brain involved at all. The skin was seeing, not the way an eye sees. There is no lens, no focused image, no ability to look at a distant object and resolve its details. The skin's light sensing is local and low resolution. Each patch of skin detects what kind of light is hitting that patch right now, but across an entire body surface covered in those local detectors, something remarkable may emerge. A distributed sense of the color of the immediate environment, assembled patch by patch, without any single organ processing the whole picture. This is the leading candidate for how a colorblind octopus produces color-accurate camouflage. The skin may not need the brain to tell it what color the nearby rock is. The skin touching that rock may be sensing the light bouncing off it directly, patch by patch, and adjusting each chromatophore based on local information alone. No central command, no color vision required. The camouflage emerges from thousands of independent local reactions happening at the same time, coordinated by physics rather than by thought. That would explain the speed. A signal from the eye to the brain to the skin would take time, however brief. A signal from the skin to the skin takes essentially no time at all. The skin self-organizes its response because each patch of it is reacting to its own local light environment in real time. Scientists are careful to note that this is still a hypothesis being tested. The skin contains the opsins. The skin reacts to light independently. Whether that reaction is what drives the full camouflage system in living animals or just a piece of it is still under active research. But even if the skin vision hypothesis turns out to be only part of the answer, the discovery itself is already extraordinary. An animal whose entire body surface functions as a distributed, low-resolution sense organ is something no one predicted before they found it. And the eyes themselves, the ones that started this mystery, have their own equally confounding trick built into their shape. Look at an octopus eye and you will notice something immediately wrong. The pupil is not round. It is not a slit like a cat's eye. It is a W. A horizontal, wavy bar, sometimes described as a dumbbell shape or a crescent, depending on the species. It is one of the most distinctive and immediately alien features of the animal's face. That shape is not an accident. Scientists think it might be the second mechanism the octopus uses to see a color it technically cannot see. Here is the physics. When light passes through a lens, different colors bend at slightly different angles. Blue light bends more than red light. This means blue light comes into focus at a slightly different distance behind the lens than red light does. In a camera, this creates an annoying problem called chromatic aberration. You have seen it as the color fringe that appears around objects in cheap photographs. The octopus may have turned that defect into a feature. Because the W-shaped pupil lets light enter the eye from multiple positions and multiple angles simultaneously, each color of light comes into focus in a slightly different place on the retina at the same time. The brain cannot detect which color it is, seeing it only has one type of photoreceptor, so it cannot separate the colors the way a human brain does. But it can detect which areas of the retina are in sharpest focus. And because different colors focus at different distances, sharp focus in one zone means the light hitting that zone is a specific wavelength. In other words, the octopus may be decoding color, not by seeing it directly, but by using focus differences as a proxy. It is reading color information through blur. A team from the University of California, Berkeley, and Harvard ran calculations in 2016, suggesting this mechanism could actually work well enough to explain the octopus's camouflage accuracy, at least in shallow water. The water itself filters out the red end of the color spectrum within the first few feet of depth, So the range of colors the octopus needs to match in its actual environment is narrower than the full visible spectrum. This means the octopus may only need to distinguish between blues, greens, and the yellows in between, a much smaller challenge than matching the full rainbow, and the W-shaped pupil plus chromatic aberration together may be enough to do it. Two separate systems, the skin opsins and the strange pupil geometry, working in parallel to solve the same impossible problem from two different angles. And these are just the eyes and the skin. The mechanism the octopus uses to change its texture adds yet another layer of physics to the already overwhelming display. Stop and picture what 230 color cells per square inch actually means. A square inch is roughly the size of a postage stamp. Pack 230 individual color cells into that space, and you have a resolution finer than most digital screens at normal viewing distance. Now cover an entire octopus body, arms included, with that density of cells, and you have a display system running into the hundreds of thousands of individual units. Each unit is a chromatophore, a tiny elastic sac of pigment connected to its own set of muscles and its own dedicated nerve connection from the brain. Every single one of those hundreds of thousands of chromatophores can be controlled independently. Every one can be expanded to full color, collapsed to invisible, or held anywhere in between. The color palette available from the chromatophores alone is yellow, orange, red, and brown, the warmer end of the spectrum. That is not the full range the octopus can produce. Underneath the chromatophores sit the iridophores, thin-plated reflective cells that produce blues and greens through structural color, meaning they create color through the physics of light reflection rather than through pigment at all. The combination of pigment-based chromatophores above and physics-based iridophores below gives the octopus a color range that spans most of what a human eye can see. New chromatophores grow throughout the octopus's life, and here is where it gets genuinely strange. Every new chromatophore that forms starts out yellow. Over a period of days, it transitions to orange, then eventually to brown. The octopus's skin is not a static tool. It is a living display that is constantly renewing, with new cells being born and maturing, while older cells are already doing their job. At any given moment, cells at different stages of maturity are all operating in the same patch of skin. How the octopus keeps track of which cell is which color, and accounts for that when assembling a pattern, is a question that has not been fully answered. The speed of the whole system is what makes it almost impossible to watch without feeling like you missed something. A full color change from one complex camouflage pattern to a completely different one can happen in under 1/10 of a second. That is faster than a human eye can process as a transition. You see one pattern, you blink, and a different one is there. In laboratory experiments, researchers have filmed octopuses moving across different colored substrates from sand to gravel to coral to rock, and each transition produces a new, accurate camouflage pattern within fractions of a second. The animal does not pause to assess. It does not freeze and recalculate. The update is continuous, flowing, instant. The mathematics of that system, the number of cells, the number of signals, the precision of the output, should require a processing demand that overloads the available neural hardware. Scientists are still working out how the octopus manages the computational load. The texture control adds another entire dimension to what the skin is doing. Changing color is one thing. Changing shape is another. The octopus does both simultaneously in under a second. Running beneath the skin are thousands of tiny muscular structures called papillae. Think of them as miniature mountains. The octopus can raise or flatten at will. In a relaxed state, they lie flat and the skin looks smooth. When the muscles contract, they shoot upward into pointed peaks, transforming the skin surface from a flat sheet into something that looks like the rough, uneven texture of rock or coral. A single papilla takes less than a second to raise or lower. Across the entire body, they can be grouped and controlled in regional patches, So the animal can make one section look rough, while another stays smooth, matching the specific texture pattern of whatever it is sitting against. This is how an octopus disappears against a jagged rock face. Changing color to match the brown and gray of the rock handles the visual tone. Raising papillae in the pattern of the rock's surface handles the three-dimensional texture. The combination breaks up the silhouette so completely that even when you know the octopus is there and you are staring directly at it, your eyes keep sliding off it. Marine biologists, studying camouflage in the field, have reported walking past octopuses they were specifically looking for because the animal's surface texture so perfectly matched the surrounding coral that the brain rejected the possibility that it was a living thing. The control of papillae appears to be driven primarily by vision. Experiments with cuttlefish, close relatives of the octopus, showed that animals raised without visual experience of textured environments do not develop normal texture camouflage. They see the texture of the environment with their eyes and use that visual signal to direct the papillae pattern. This creates an interesting loop. The octopus sees the texture of what it is sitting on. It adjusts its own surface texture to match, but it is colorblind and may be using its skin to sense the color, not its eyes. Two different sensory systems, visual and skin-based, are handling different parts of the same camouflage task simultaneously. The result is so convincing that it works against predators whose color vision is sharper than a human's. And the octopus does not just hide from its enemies. One species has evolved an approach so bold, so counterintuitive, that when researchers first filmed it in the wild, they assumed they were seeing a mistake. In 1998, a diver off the coast of Sulawesi, Indonesia, filmed an octopus doing something no animal had ever been recorded doing before. The octopus gathered its arms together, flattened its body into a thin oval, and began gliding across the seafloor in a slow, undulating wave motion. It had turned itself into a flatfish, a toxic flatfish, the kind that local predators avoid. Then the same animal, a few minutes later, rearranged its arms into a fan around its body and drifted upward, trailing them like venomous spines. It had become a lionfish. Then it twisted two arms into a tight spiral, held the others out, and moved like a banded sea snake. One animal. Three different animals, all within a single crossing of open seafloor. This is the mimic octopus, formally discovered and named in 2005, and it is still the only known animal that actively impersonates multiple different species, selecting its disguise based on which predator is nearby. It can imitate at least 15 different animals, all of them venomous, toxic, or otherwise dangerous to eat. The list includes sea snakes, lionfish, flatfish, jellyfish, sea anemones, mantis shrimp, stingrays, and brittle stars. Each imitation uses a different combination of arm position, body shape, movement pattern, and color display. The selection is not random. When a damselfish threatens the mimic octopus, damselfish are aggressive towards sea snakes. The octopus mimics a banded sea snake. When a different predator approaches, it switches to a different imitation, specifically chosen for what that predator avoids. This requires holding a mental catalogue of which predator fears which animal and making a real-time selection based on visual identification of the approaching threat. No other animal on Earth is known to do this. The evolutionary path to this behavior is even stranger. DNA analysis shows that the mimic octopus's ancestors first developed bold, high-contrast black and white markings as a general shock defense. Then they developed the flatfish swimming motion. Then, finally, they began combining color, movement, and body position into fully committed impersonations. The deception evolved in stages, each stage building on the last, towards something with no parallel in the animal kingdom. The animal that hunts in disguise and hides in disguise and impersonates its enemy's worst fears while crossing open water is already at the edge of what seems possible. What the octopus does with objects it finds on the seafloor crosses that edge entirely. In 2009, a team of researchers filming octopuses in the shallow ocean off Indonesia watched something that stopped them cold. A small octopus was moving across a flat, open patch of sandy seafloor, exactly the kind of exposed ground where octopuses are most vulnerable. As it moved, it reached down, picked up two halves of a coconut shell, and tucked them underneath its body. Then it kept walking. Or more accurately, it kept stilt walking, moving awkwardly on its arm tips with the shells clutched beneath it, slower than normal, more exposed, using more energy than simply crawling. 20 feet further on, it stopped, arranged the two shell halves together into a dome, and climbed inside. The shells were shelter. The octopus had carried them as a portable shelter, accepting the cost of awkward, exhausting stilt walking across exposed ground, because it had calculated, or something functionally equivalent to calculated, that the shelter would be worth having at the destination. Carrying a tool to a different location for use in the future requires imagining a scenario that does not yet exist. It requires valuing an object not for what it does right now, but for what it will do later. Until this discovery, that capacity for future-oriented tool use had only been documented in great apes, corvids like crows and ravens, and humans, a group so exclusive it was practically written into the definition of what complex cognition means. Coconut shells were introduced to the Indonesian ocean floor by humans relatively recently. The octopus did not evolve over millions of years to use coconuts specifically. It recognized the potential of a new object and invented a use for it. That is innovation, applied to a tool it had never encountered before. The same octopus species has been observed using rocks as doors. It enters a crevice, pulls rocks and shells toward the opening until the entrance is mostly sealed, and sits behind its self-built wall. If disturbed, it adds more material. It is building a barricade out of available objects, adapting the barrier based on the size of the threat. These are not instincts playing out on a fixed script. Instincts do not flexibly adapt to new materials. Instincts do not plan for future shelter at a location that does not yet exist when the plan forms. What the octopus is doing requires something much more flexible than that. And it is doing this with a brain the size of a walnut in a lifespan shorter than most school years. What it does in captivity, surrounded by humans, is even harder to explain. In 2016, an octopus named Inky made international news by escaping from the National Aquarium of New Zealand. Staff arrived one morning to find his tank empty. A small gap had appeared in the lid overnight. The gap was wide enough for a tennis ball. Inky's body, when relaxed, is soft enough to flow through any opening, larger than his beak, a hard, parrot-like structure, roughly the size of a fingernail. He squeezed through the gap, dropped to the floor, crawled across the room, and found a drain pipe. The drain pipe led to the ocean. He was gone. This was not the first time something like this happened, and it was not the last. Aquarium records going back over 100 years document octopuses escaping tanks, raiding neighboring tanks for food, and in several documented cases, returning to their own tanks before staff arrived in the morning. One aquarium in Brighton, England, noticed their population of small fish declining across multiple tanks with no explanation until a keeper stayed late and watched an octopus leave its enclosure, travel between tanks, eat, and return home. These escapes are not random thrashing. They are planned. The octopus observes its environment over days or weeks, identifies weak points in the enclosure, selects a moment to act, usually overnight when the aquarium is dark and quiet, and executes a multi-step escape sequence. Each step requires the previous one to work. The octopus has to know where the gap is, how to reach it, what is on the other side, and which direction leads to water or food. Several aquariums have documented octopuses learning to short-circuit their tank lights by squirting precisely aimed jets of water at the electrical fixtures. They did this repeatedly, and for no apparent reason other than that the light bothered them. The precision required to hit a small target with a water jet repeatedly and consistently while inside a tank from a moving position is not trivial. Some aquariums have installed elaborate multi-lock systems specifically to contain octopuses, The animals routinely defeat the first generation of locks. Scientists who study captive octopus behavior have described them as the most consistently difficult animals to contain, not because they are physically powerful, but because they investigate their environment systematically and remember what they find. The same animals that plan escapes in the dark also recognize the faces of the specific humans they dislike. Scientists at a university in Seattle ran an experiment with captive octopuses and two specific staff members. One staff member fed the octopuses every day, handled them gently, and gave them consistent positive attention. The other staff member poked them with a stiff bristle brush during checkups, an irritating but harmless stimulus designed to create a mildly unpleasant association. Within a few weeks, the octopuses had sorted the two humans out completely. When the feeding staff member approached, the octopuses moved toward the front of the tank, showed active color displays, and extended an arm toward the glass. When the bristlebrush staff member approached, the octopuses moved away, darkened in color, and in multiple cases directed a jet of water at the person from a distance. The two staff members made the same general sounds, the same general size and shape movements. The octopuses were distinguishing them by face alone. Individual face recognition in animals is typically associated with species that have strong social bonds and long-term relationships. Recognizing a specific individual matters when you will see them again and remember the history. For a solitary animal with a 1 to 2 year lifespan, developing the neural hardware for face recognition seems like an expensive solution to a problem that barely exists. An octopus encounters only a handful of specific individuals over the course of its entire life, but the recognition goes further than identification. The octopuses were not just telling the two humans apart, they were applying stored emotional valence to each one, directing affection toward one and active hostility toward the other. The behavioral response was not neutral identification, it was clearly charged. At the Seattle Aquarium, keepers documented an octopus that disliked one specific volunteer so intensely that every time that person passed the tank, the animal surfaced and sprayed water at them. Other volunteers walking the same path produced no reaction. The octopus targeted the specific person every time across weeks. What drives this level of behavioral specificity in an animal this structurally different from us is an active research question. The octopus brain has no direct analog to the regions humans use for face processing. Whatever it is doing it a completely different way. The individual personality behind those responses adds another layer entirely. For decades, the scientific consensus was that invertebrates do not have individual personalities. Individual differences in behavior, shyness, boldness, aggression, curiosity were thought to require complex social structures and long-term social memory to make them evolutionarily useful. An animal with no lasting relationships, no social group, no reason to track how individuals differ from each other would have no use for a personality. And without evolutionary pressure to produce personality would not arise. The octopus broke that rule cleanly. A study published in 2023 in the journal Current Biology tested octopuses on a battery of behavioral tasks and measured how individual animals responded. Some octopuses approached new objects immediately, explored them thoroughly, and solved problems faster, even when the approach increased their short-term risk. Others hung back, assessed from a distance, and moved more cautiously. These patterns were consistent across different types of tasks, different days, and different testing conditions. The same octopus that was bold in an exploration task was bold in a food access task. The same octopus that was cautious once was cautious again. That consistency across contexts is the scientific definition of personality, and it appeared reliably in a solitary, short-lived invertebrate with no social structure. The researchers found something else. Boldness and problem-solving speed were not the same trait. Some octopuses were bold and solved problems quickly. Others were bold but worked slower and more deliberately. The cognitive traits were separable. Each animal carried its own combination, its own cognitive fingerprint. Every octopus was, in some measurable sense, genuinely different from every other octopus. Aquarium keepers have known this informally for a long time. Specific animals develop reputations. This one is curious and will investigate anything new. That one is shy and hides for a week before it trusts its environment. Another one is hostile. Another is playful. The keepers who work with the animals daily describe them with the vocabulary of character because nothing else fits what they observe. A 2025 paper in the journal Trends in Ecology and Evolution extended this further, presenting evidence that octopuses can engage in deliberate behavioral deception, tricking other organisms through intentional misleading behavior. This cognitive ability had previously been documented almost exclusively in primates and in the most cognitively advanced bird species. Individual personalities, face recognition, deception, and the animal that has all of this lives for 1 year. An octopus had never seen a jar before. A second octopus had watched another octopus open one. When both were tested on the same jar, the one with no prior experience took an average of about 5 minutes to open it. The one that had watched another animal do it took less than a minute. That experiment, run across multiple trials and multiple animals, confirmed something extraordinary. Octopuses can learn by watching. They observe a behavior performed by another individual and use that observation to improve their own performance on the same task. Observational learning is a specific cognitive skill with a specific requirement. The observer has to mentally represent what the other animal is doing, understand the goal behind the action, and then map that observed sequence onto its own body to replicate it. It is not simply repeating a sound or following a scent trail. It is watching purposeful action and extracting the logic of it. This kind of learning was long considered a feature of animals with dense social lives, primates who live in complex groups, crows who teach their young, dolphins who watch their mothers hunt. The assumption was that observational learning evolves because it is useful inside a social structure, where copying a group member's behavior gives an individual a shortcut to tested knowledge. The octopus has no group. It has no young it teaches. It almost never encounters another octopus except to mate, and even that encounter tends to be brief. The social pressure that was supposed to drive this ability does not exist for it, and yet it watches. It stores what it saw. It applies it. One documented case at the New England Aquarium showed an octopus opening a child-proof pill bottle after watching a researcher demonstrate the technique once. Child-resistant containers are specifically designed to defeat unthinking force. Opening one requires a non-obvious sequence of moves. Press down while turning in a specific direction at a specific angle. The octopus watched the researcher perform this sequence one time and replicated it one time. The cognitive load of that task, watching an action performed by a different type of body, extracting the underlying physical logic, and then translating it into a completely different set of limbs is substantial. Humans learn from watching once. Smart birds learn from watching once. Octopuses apparently do too, from an evolutionary branch that split from ours before watching another individual's hands had any meaning at all. What it learns in its brief life, it carries without anyone to teach it and no teacher to learn from. And the life it carries all of this in is far shorter than it has any right to be. An octopus lives 1 year, some species push to 2. The giant Pacific octopus, the largest of all, can reach 4. But most of the octopuses studied for their intelligence, the ones opening jars, escaping tanks, recognizing faces, learning from observation, are gone within 12 to 18 months of being born. Compare that to every other highly intelligent animal on the planet. Dolphins live 40 to 50 years. Elephants live 60 to 70. Crows live up to 30. Chimpanzees live over 40. Even the relatively simple-brained parrot outlives the octopus by decades. The pattern in the animal kingdom is consistent and almost universal. Big brains take time to pay off. Intelligence is expensive to build and expensive to run. A brain this complex, one that can learn, remember, problem solve, and adapt, only makes evolutionary sense if the animal has enough time to use what it learns. An elephant uses its remembered knowledge of water source locations during famines that may occur once every decade. A crow teaches its young techniques it spent years developing. A chimpanzee builds social alliances over years that eventually determine its access to food and mates. All of that requires time. The payoff from intelligence scales with lifespan. The octopus breaks this rule so completely that it has forced scientists to invent a new theory to explain it. Everything a highly intelligent animal typically does, the octopus does alone. It cannot rely on a social group to share what it learned. It cannot teach offspring. It cannot build on knowledge across generations the way social species do. Whatever it figures out by itself, and it uses what it learns within the single year of its existence, and then it is gone. And the thing that ended its life was written into its biology before it was even born. The octopus does not die of old age. It dies on a schedule. The male dies after mating. The chemistry of reproduction triggers a biological process called senescence, a controlled shutdown of the body's maintenance systems. He stops eating. He stops healing. Injuries that would have closed quickly a few weeks earlier now stay open. His behavior becomes disorganized. Within days to weeks of mating, he is dead. The female survives slightly longer. She finds a sheltered space, deposits her eggs in strings attached to the ceiling of her den, and then stops doing almost everything else. She stops hunting. She stops eating. For weeks or months, the duration depends on the species. She devotes all remaining function to protecting the eggs. She fans them constantly with her arms to keep fresh, oxygenated water flowing over them. She keeps predators away. She does not leave. When the eggs hatch, she typically dies within days. The eggs she protected contain no parental knowledge. Octopus hatchlings receive nothing from their mother beyond the genes. No lessons, no guided first hunts, no model to watch. They emerge into the open ocean as tiny, fully autonomous individuals who must figure everything out alone. And they do, which raises the question that has kept marine biologists arguing for years. Why did evolution build this level of intelligence into a system so clearly designed to disappear so fast? The standard theories of intelligence, evolution, social complexity, parenting demands, long-term learning, all point toward longer life and social structure, neither of which the octopus has. Something else drove the octopus to this brain. Something about the specific demands of its environment made intelligence the winning strategy, even without a long life to use it in. That answer exists, and it points to something about the nature of intelligence itself that no one expected from an animal this brief. The octopus lives 1 year, and that 1 year is spent in one of the most demanding environments any animal can occupy. To understand why intelligence evolved under those conditions, picture what an octopus actually faces every day. It has no shell, no armor, no speed advantage over most of its predators. Its body is soft enough to be eaten in a single bite by a moray eel, a shark, a large fish, or a seabird if it surfaces. It hunts animals that fight back, crabs with claws, shrimp that can launch backward at speed, fish that bite. And it lives in an ocean where the terrain changes constantly. Shifting sand, rearranged rocks, coral that looks different from every angle, prey that hides in gaps too small to see from the surface. Every single meal requires a fresh decision. Every escape from a predator requires a new solution. There are no fixed rules for how to survive an octopus's daily life. The environment is too variable. The threats are too diverse. This is the ecological pressure theory, and it argues that the octopus did not evolve intelligence the way social animals did by learning from a group over a long life, but instead evolved it as an individual survival tool for navigating a wildly unpredictable world, one problem at a time, in real time. Intelligence in this model is not about accumulating knowledge over decades. It is about being capable enough in the moment to handle whatever the current moment throws at you. A chimpanzee's intelligence is partly about social alliances and group memory built over 40 years. An octopus's intelligence is about solving this specific crab in this specific crevice problem right now, today, with no reference to what worked last time and no one to ask. The brain that serves that purpose does not need a long time to pay off. It needs to work well immediately, every time, for a brief and brutal year. After that year, it passes its genes to the next generation and disappears. The genes for that level of intelligence persist because the animals that carried them survived long enough to reproduce. The ones without them died in the crab's claws or the eel's mouths before they ever got that chance. The octopus is the proof that intelligence does not require time, social structure, or a long return on investment. It just requires sufficient pressure, and below the surface of that brief, pressured life, the octopus is doing something with its own molecular code that no animal on Earth does at anywhere near the same scale. Most animals adapt to their environment over generations. A population lives in cold water for thousands of years, and the individuals best suited to cold water survive more often and pass on their genes. Over time, the population shifts. The adaptation is baked into the DNA itself. The octopus does something faster. When the water temperature drops, it rewrites its own brain chemistry within hours. Every cell in every living thing operates by reading instructions from DNA, copying those instructions into molecules called messenger RNA, and then using the RNA as a blueprint to build proteins. The proteins do the actual work inside cells, running chemical reactions, transmitting signals, maintaining structure. Normally, the step between DNA and protein is fixed. The DNA says build this protein, the RNA copies that instruction exactly, and the protein gets built as written. The octopus intercepts the RNA step and edits it. Specialized enzymes in the octopus's nervous system reach into the copied RNA message and swap out individual chemical letters, changing the instruction before it reaches the protein-building machinery. The result is a slightly different protein from a gene that did not change at all. The DNA stays the same, but the protein the cell actually produces is altered on the fly. In 2023, a team at the Marine Biological Laboratory in Massachusetts documented this system in detail. When they cooled octopus tanks by about 18 degrees Fahrenheit, the animals responded within hours by editing more than 13,000 different RNA sites in their nervous systems. A third of their neural protein instructions changed. The proteins that conduct nerve signals, the ones that control how quickly an arm moves, how fast a sucker grips, how reliably a neuron fires, all got modified to work efficiently at the new temperature. The same brain that was operating in warm water switched over to a cold water optimized version at the molecular level, without any change to the underlying genes. In humans, adjusting to temperature happens slowly, over weeks, primarily through changes in blood flow and metabolic rate. In the octopus, the brain literally upgrades its own hardware in a matter of hours by rewriting the molecular instructions it is reading. This is not how biology was supposed to work. Animals were supposed to adapt across generations, not across hours. The octopus collapsed that timeline to something closer to software, updating itself mid-run. And the genome that enables all of this is, on paper, even stranger than the system itself. When scientists sequenced the complete octopus genome in 2015, they expected to find an interesting invertebrate genome. What they found looked like someone had put a normal animal genome in a blender, hit the button, and poured out the result. The California two-spot octopus has 33,000 protein coding genes. Humans have around 20,000. That alone puts the octopus comfortably above us in raw gene count, which is already a strange sentence to write about an animal that fits in a coffee mug. The additional genes do not appear to be duplicates of existing ones. They include hundreds of genes found only in octopuses and their closest relatives' genes with no known equivalent in any other animal on Earth. What those genes do is still being worked out. Some are expressed specifically in the brain. Others are active in the skin and suckers, they appear to be doing things that no other genome needed to do. The arrangement of the genes is the part that made the research team use the phrase, "put in a blender." In almost every animal, genes that work together to control the same biological process cluster together in the same region of the same chromosome. They stay neighbors over hundreds of millions of years of evolution because keeping them close helps them coordinate. This clustering is so universal that it has been used to trace animal evolutionary relationships for decades. In the octopus, those clusters are gone. The genes that control body plan development, called Hox genes, found in the same organized block in almost every bilateral animal, are scattered randomly across multiple chromosomes with no apparent logic to their positions. The genes that work together to build the nervous system are spread across the genome with no apparent grouping. The whole genome looks, as one researcher on the team put it, like it just does not follow the rules that the rest of the animal kingdom agreed on. What drove that reshuffling is still an active question. One candidate is the activity of jumping gene segments of DNA that can copy themselves and land in new locations, disrupting the surrounding neighborhood each time they move. The octopus genome has a very high density of these mobile elements, and their activity appears to be highest in the brain and nervous tissue. Scientists suspect the reshuffling itself may be part of how the octopus generates neural complexity by constantly moving regulatory elements into new positions near existing genes and producing new patterns of expression. A genome scrambled for 500 million years, editing its outputs in real time, running 9 parallel brains across a boneless body, dreaming in color. That last one deserves its own chapter. In 2019, a marine biologist sat with a camera in front of a tank and watched an octopus named Heidi fall asleep. Heidi turned pale, curled her arms loosely, and went still. Then, about half an hour later, something else began. Her skin started flickering. Pale cream shifted to orange-brown. Patterns bloomed across her mantle, the textures of coral, the mottled shades of sand cycling through one vivid image after another, while her arms twitched and her eyes moved beneath closed lids. Heidi was asleep the entire time. The footage went global. Scientists watching it described the skin changes as identical to what they saw in octopuses, actively camouflaging against real environments. The pattern sequences looked like a highlight reel of camouflage routines. If a human moved their hands while sleeping in the exact sequence they used to play a specific song on piano, you would suspect they were dreaming about playing that song. Since then, formal research has confirmed that octopuses experience 2 distinct sleep stages. The first is quiet sleep, pale, still, with slow, regular neural activity resembling what is called sleep spindles in human non-dream sleep. Scientists think sleep spindles are involved in processing and storing memories. The second stage is active sleep. About every 30 to 40 minutes, the quiet phase breaks. The octopus's skin erupts in color, its arms twitch, its eyes move, its breathing speeds up. The neural activity recorded in this phase matches the brain patterns seen when the octopus is awake and actively doing things. This active sleep stage lasts only around 1 minute, then it ends and quiet sleep returns. Researchers at the Okinawa Institute of Science and Technology confirmed in 2023 that the brain activity during active sleep is not random noise. It tracks specific patterns tied to the animal's behavior. The skin patterns that appear during active sleep mirror the specific patterns seen when the octopus was awake and hunting or hiding. No one can ask an octopus if it is dreaming. The answer may never be certain, but the evidence suggests that some replay of experience, some consolidation of the day's hunting and hiding, is happening in that sleeping body while the colors flicker across it in the dark. One of the most alien minds on Earth may be spending its brief life dreaming. A moray eel bites an octopus arm and pulls it free. The octopus jets away, 7-armed now, bleeding from the stump. But the arm on the seafloor does not stop. For up to an hour after separation, the arm continues moving. It reacts to touch. It explores surfaces with its suckers. It passes objects toward where the octopus's mouth used to be. The local nervous system in the arm keeps executing its last known program: reach, taste, grip, feed, with no body attached. This is not reflex. A reflex is a single, fixed response triggered by a single stimulus. What the severed arm does is a sustained, goal-directed behavioral sequence running on the arm's own neural hardware, long after the central brain has left the building. Each octopus arm contains hundreds of thousands of neurons arranged in a dense nerve cord running from the base to the tip, with local branches at every sucker. That cord carries enough processing power to run the arm's basic functions independently. The central brain normally coordinates the arms by setting goals and monitoring outcomes. But the arms do not need the central brain to act. They need it to stop them from acting on their own. This has a remarkable consequence for surgery and regeneration. Young octopuses can regrow a lost arm, entirely nerves, muscles, suckers, and all in a matter of weeks. Adult octopuses lose that regenerative ability as they age, but the regrown arms in juveniles function normally, with a fresh nerve cord and working suckers, as if the system was simply reinstalled from a backup. The arm growing back is not rebuilding from scratch, using a stored blueprint. The local nervous system at the stump seems to guide the reconstruction process, signaling what the arm should look like and sending growth factors outward along the developing tissue. The new arm grows into the correct shape because the biological information for building that shape is already present in the remaining stump tissue. Robotics engineers have spent years trying to build arms that can handle unpredictable objects in unstructured environments. The octopus did it with a nerve cord and a set of suckers, and left behind enough of a blueprint that a new arm can regenerate itself from a stump. The engineering problem these animals solved 500 million years ago is still unsolved on any drawing board on Earth. The tiniest species in this lineup has done something the engineers haven't even begun to crack. The blue-ringed octopus fits inside a coffee cup, fully grown. It weighs less than an apple. Its arms span about 8 inches across, smaller than a spread human hand. It is a quiet, slow-moving animal that spends most of its time wedged into small crevices in shallow reefs across the Pacific and Indian Oceans. It carries enough venom to kill 26 adult humans. The venom is called tetrodotoxin. It blocks the channels in nerve cells that allow sodium to move, shutting down the nerve's ability to fire a signal. The effect spreads through the body in minutes. Muscles stop responding. The diaphragm, the muscle that drives breathing, stops. A person bitten by a blue-ringed octopus can be fully conscious, completely aware of what is happening, while their body loses the ability to breathe. Without immediate mechanical ventilation, death follows in minutes. There is no antivenom. The only treatment is keeping the person breathing artificially until the toxin clears, which can take hours. The octopus does not produce the toxin itself. Bacteria living in its salivary glands produce it, and the octopus concentrates and deploys it through a bite from its small, hard beak. The same bacteria produce tetrodotoxin in pufferfish and certain other poisonous ocean animals. The toxin evolved independently in different species as a defensive tool, but the blue-ringed octopus carries it in quantities far beyond what its body size suggests possible. The warning is written on its skin. Under normal conditions, the blue-ringed octopus shows muted browns and yellows. When threatened, the rings hidden in folds of its skin contract, exposing vivid, iridescent blue circles across its body. The blue rings are not pigment, they are structural color, produced by layers of reflective cells stacked like microscopic mirrors. They flash with an intensity that is visible even in murky water. The flash means leave now. Every predator that has ever learned this lesson passed that lesson on. The smallest, most venomous, most visually spectacular octopus on Earth is also the one whose cardiovascular system reveals the body's deepest engineering compromise. Swimming kills an octopus. Not literally, not immediately, but each time an octopus pushes water through its siphon to jet through the water column, The third heart, the one that drives oxygenated blood through the body, stops beating. The effort of propelling the body shuts down the main blood pump. The octopus is, in a meaningful physiological sense, running on reserves the moment it leaves the seafloor. This is why octopuses almost never swim long distances. Short bursts to escape a predator. Yes, they can accelerate faster than almost any animal their size for a few seconds. But sustained swimming exhausts them at a rate that would be alarming if you were watching their internal organs instead of their graceful movement. It is also one of the stranger design compromises in all of animal biology. The octopus has 3 hearts specifically to handle the demands of a complex, high-metabolism body. 2 push blood through the gills to collect oxygen. The third, the systemic heart, takes that oxygenated blood and drives it to the muscles, brain, and organs. The system is efficient at rest. It falls apart the moment locomotion requires jet propulsion because the muscular contraction that propels the jet physically interferes with the systemic heart's ability to contract. Evolution appears to have decided that the octopus should crawl rather than swim, and built the heart around that compromise. Crawling is slower but preserves full cardiovascular function. Swimming is fast but pays for it immediately. The result is an animal that moves with extraordinary control and efficiency along the seafloor, exploring, hunting, hiding, building shelters, investigating objects, and makes brief, explosive, heart-stopping dashes through open water when something forces it to. The blood those hearts pump is blue, carried by molecules built around copper rather than iron, designed specifically to offload oxygen efficiently in cold, low-pressure environments. The whole system is a set of solutions stacked on each other, each one solving a problem created by a different part of the same body. And when that body needs to escape fast, and camouflage has already failed, and the arms are no help, it fires one more weapon, one that works on senses the predator did not know could be attacked. The octopus releases its ink and the water goes black. Most people assume that is the whole trick. Smokescreen. Predator confused. Octopus escapes. Simple. The ink does something far more targeted than that. Octopus ink contains a compound called tyrosinase. When it disperses through the water, tyrosinase binds to the olfactory receptors of any animal swimming through the cloud. Olfactory receptors are the sensory cells used for detecting chemicals in water in fish and sharks. They are the primary hunting tool, far more important than sight for tracking prey across any meaningful distance. Tyrosinase temporarily disables them. A shark hunting by scent inside an ink cloud loses the ability to smell. An eel tracking chemical, traces left by the octopus's movement, hits the cloud and loses the chemical trail entirely. The ink does not just block their vision, it erases the sensory data they rely on most. The octopus also deploys 2 structurally different types of ink release simultaneously. The first is a diffuse cloud, a broad spreading haze that disperses through a large volume of water and creates the general smokescreen effect. The second is a pseudomorph, a dense, compact blob of ink held together by mucus that maintains roughly the octopus's shape and size for long enough to fool a visual predator into attacking it while the real animal jets away in the opposite direction. The pseudomorph is a decoy. It has the approximate silhouette of an octopus, suspended in the water where the octopus just was, while the octopus itself vanishes, changes color, and presses flat against the seafloor, 15 feet away. A simultaneous multi-target attack on the predator's vision, its sense of smell, and its ability to track a specific target deployed in under a second, from a gland the size of a thumb, by an animal with no bones, and the molecule that produces all of this melanin is the same pigment that gives human hair and skin their color. The body that produces all of this runs on a genome so unusual that the first scientists to fully read it reached for the word they had been trained never to use lightly. When Caroline Albertin, a graduate student at the University of Chicago, helped sequence the first complete octopus genome in 2015, her team sat down to compare it against the genomes of other animals. They expected to find an unusual invertebrate genome, What they found required a different framework entirely. In her published notes, she wrote that the octopus genome looks like it was put into a blender and mixed. She meant that precisely. In almost every animal on Earth, the genome has a structure. Genes that work together stay together on the same chromosome, in the same region, close to each other. This arrangement is called synteny, and it is conserved across hundreds of millions of years of evolution because it works. Coordinated genes produce coordinated outcomes. The genomes of flies and worms, and fish and humans, all share blocks of synteny that trace back to their last common ancestors. The octopus genome has almost none of it. The genes that control how the octopus builds its body are scattered across the genome with no apparent order. The genes that coordinate development of the nervous system are on different chromosomes from each other. The whole arrangement looks like the result of some massive disruption event, as if a force moved hundreds of thousands of DNA segments to random new positions over a very long time. That force appears to be jumping genes, called transposons, segments of DNA that can copy themselves and insert the copies into new locations within the genome. Nearly half of the octopus genome consists of these mobile elements, and their activity is highest in the brain and nervous tissue. Every time a transposon lands in a new location, it can drag nearby regulatory signals with it, placing genes under new controls and potentially generating new patterns of expression. The team's working hypothesis is that the genomic chaos is not a side effect of octopus evolution, it is a driver of it. By constantly reshuffling regulatory elements near brain-expressed genes, the jumping genes may have produced new neural capabilities faster than normal evolution would allow. An accelerated molecular experiment running for hundreds of millions of years, generating combinations of gene expression that no other lineage ever tried. It is a genome built for innovation through disorder, and it produced a brain that, while sleeping, does something almost no one predicted was possible in an invertebrate. An engineer trying to design a robotic arm faces a fundamental problem. A rigid arm made of metal joints and servo motors can be controlled precisely because every joint bends in exactly one direction by a known angle in response to a known input. The math is straightforward. The outcome is predictable. A soft arm made of continuous flexible material with no joints can bend in any direction at any point along its length, which means it has essentially infinite possible positions. Controlling it requires solving an enormous number of variables simultaneously, faster than any central processor can handle if you route every micro-decision through a single brain. The octopus already solved this in a completely different way. The local nerve cords in each arm handle the moment-to-moment control. The central brain sets the goal and monitors the outcome. By distributing the decision-making, the octopus eliminates the central processing bottleneck. The arm reaches around a corner, finds an object, and wraps around it without the brain ever computing the exact configuration of each point along the arm. Engineers at the University of Illinois built the Cyber Octopus project, funded by the US Navy's Office of Naval Research, to model and replicate exactly this architecture. They built a computational model of an octopus arm with all of its muscles accurately represented, about 200 individual muscles per arm, and then developed control algorithms that mimic the distributed processing of the octopus's arm nervous system. The goal is a new generation of soft robots capable of operating in unstructured environments, reaching into gaps too tight for rigid equipment, handling objects of any shape without pre-programming a specific grip, navigating around obstacles by feel rather than by mapping. Current rigid robotic arms require the environment to be structured around them. An octopus architecture arm can adapt to whatever it finds. Research papers on octopus-inspired robotics grew from 760 in 2021 to over 1,100 in 2024, a 54% increase in 3 years. The engineering world is moving fast toward the blueprint the octopus has been running for 500 million years. And the most powerful country in the world has decided this animal's nervous system is worth funding at the level of a military research initiative. The Office of Naval Research does not usually fund research into marine biology for its own sake. When it committed funding to the Cyber Octopus project at the University of Illinois, the goal was clear: understand how the octopus's distributed intelligence works well enough to replicate it in machines. The specific capability they wanted to crack was autonomous operation in unstructured environments without central coordination. Every military robotic system currently in use runs on centralized control. A drone has a computer that processes every sensor input and decides every movement. A robotic arm on a submarine has a processor routing every joint command. This architecture works in controlled environments with reliable communication. It breaks down in complex, unpredictable terrain where the decision speed required outpaces what a central processor can handle, or where communication with that processor is delayed or cut. An octopus's arm can reach into a crevice, find an object, assess it chemically and tactilely, decide whether it is worth grabbing, and close its suckers around it all without a round-trip signal to the central brain. For military applications, that means an autonomous underwater vehicle with octopus architecture limbs could operate in complex seafloor environments, navigate around obstacles, and interact with physical objects without requiring continuous high-bandwidth communication with a control center. The computational model built by the team successfully replicated the arm's reach and grasp behavior using distributed control algorithms. Instead of one processor computing the full arm trajectory, small local controllers at intervals along the arm each handled only their immediate section, communicating with their neighbors. The emergent outcome, a smooth, adaptive, goal-directed reach, arose from the coordination of local decisions rather than from a central plan. Biologists have spent decades trying to understand what makes the octopus intelligent. Engineers and military researchers have spent the last few years trying to steal the answer. The fact that two-stage sleep evolved independently in an animal this structurally different from us points to something even deeper about what intelligence might require. 540 million years ago, the ancestor of every vertebrate and the ancestor of every octopus parted ways permanently. Both descended from something simple, a primitive worm-like creature with light-sensitive patches of skin but nothing resembling an eye. Over the next several 100 million years, the two lineages went entirely separate directions. The vertebrate line developed a brain in the head, bones in the body, and a visual organ with a cornea, a lens, an iris, and a layered retina. The cephalopod line developed a distributed brain across a boneless body, a skin-based color-sensing system, and a visual organ with a cornea, a lens, an iris, and a layered retina, the same eye, built from different cells, guided by different developmental genes, from ancestors that had not shared a single design feature for more than 500 million years. It is the most famous example of convergent evolution in all of biology. The human eye has a blind spot. The optic nerve exits through the back of the retina, interrupting the layer of photoreceptors at that point, leaving a gap in the visual field that the brain fills in with guesswork. The octopus eye does not have a blind spot. Its photoreceptors face the incoming light directly, and the optic nerve exits from behind, leaving the retinal surface uninterrupted. In a narrow technical sense, the octopus eye is better designed than ours. Both eyes arrived at the same solution to the same problem: how to form a focused image from reflected light independently, starting from nothing, separated by 500 million years of completely isolated evolution. And they both produced a sophisticated two-layer light collection system with a variable aperture and a neural connection to a complex brain. When evolution solves the same problem the same way twice, from scratch, it suggests the solution is not coincidental. It suggests that for certain problems, and building an eye that forms a focused image is one there are only so many good answers. The universe has physical constraints. Some designs are simply better than others, and given enough time and pressure, evolution finds them. Both the octopus and the vertebrate line found the same one. That says something about the nature of physics, and about the nature of sight that no biology textbook has fully worked out yet. The fossil record of the animal that built all of this just got rewritten in a way no one expected. In the year 2000, a paleontologist looking at a 300-million-year-old specimen from a site in Illinois made a call that would stand as fact for 25 years. The fossil, a flattened blob roughly the size of a human palm, pulled from the famous Mazon Creek deposit, appeared to have a soft sack-like body, paired fins, and a cluster of arm-like structures. The researcher identified it as an octopus, named it, and published the finding. It entered the Guinness World Records as the oldest octopus ever found. The identification pushed the octopus timeline back by 150 million years. Previously, the oldest confirmed octopus fossils came from the Jurassic period, roughly 90 million years old. This specimen suggested octopuses had existed since long before the dinosaurs appeared. It became a calibration point for evolutionary timelines. Papers cited it. Models relied on it. In April 2026, a team at the University of Reading scanned the same fossil using a synchrotron, a machine that generates beams of light hundreds of times brighter than the sun, capable of illuminating the internal structure of rock without damaging it. Inside the stone, they found a ribbon of teeth, rows of 11. Octopuses have either 7 or 9 teeth per row, never 11. The animal was not an octopus. It was a decaying nautilus, a shelled cephalopod whose soft tissue had been warped and rearranged as it decomposed before fossilization, making it look like something entirely different. The Guinness World Record was wrong. The calibration point was wrong. 25 years of phylogenetic calculations that used the fossil as an anchor point need to be revisited. The honest fossil record now puts the octopus's confirmed ancestry at roughly 90 million years old, still ancient enough to predate most of what we think of as natural history, but far more recent than the animal's own genome suggests. Based on molecular clock estimates, the octopus is already harder to understand than almost any other animal on Earth. It turns out its evolutionary history is still being revised, in real time, using technology powerful enough to look inside a rock. The newest member of that evolutionary line may be hiding in water that no one has looked at yet. For most of legal history, animal welfare protection applied to vertebrates. The line was simple. Animals with backbones could potentially feel pain, and the law would protect them. Animals without backbones in vertebrates were assumed not to feel in any meaningful sense, and the law largely left them out. In 2021, a team of researchers at the London School of Economics reviewed over 300 scientific studies on cephalopod mollusks and decapod crustaceans, the group that includes octopuses, crabs, and lobsters. They applied 8 criteria for assessing the likelihood of sentience: the presence of pain receptors, connections between those receptors and brain processing regions, behavioral responses to injury, the ability to trade pain against reward, and several others. Their conclusion, the evidence for sentience in octopuses, was very strong. Not possible, not speculative, very strong. That report was presented to the UK government, which then amended the Animal Welfare Sentience Bill to include cephalopods and decapod crustaceans for the first time in history. The law passed in 2022. Octopuses became the first invertebrates ever protected under a general animal welfare statute in the UK. What this means legally is that the welfare of an octopus must now be considered in future UK policy decisions, in research, in commercial fishing practice, in aquaculture. The question of whether an animal without a spine can suffer had a legal answer in most countries for most of history. No. The octopus changed that answer. This matters beyond the octopus. The legal shift required accepting that intelligence, consciousness, and the capacity to suffer are not exclusive to the animal body plan humans are most familiar with. A creature that solves puzzles, recognizes faces, plans ahead, and possibly dreams, but does all of it with a distributed nervous system, no backbone, and blue blood forced lawmakers to stop using vertebrate anatomy as the standard for inner life. The definition of what counts as a mind, in the eyes of the law, expanded to include the octopus, and the implications of that expansion for how we think about minds in general reach far beyond the ocean floor. For most of the history of thinking about consciousness, philosophers and scientists worked with one example, the human brain. Later, they worked with primate brains, and then bird brains, and then fish brains. All of these share the same fundamental architecture: a centralized organ in the head, a spinal cord connecting it to the body, sensory inputs routed upward, and motor commands routed downward. Every model of consciousness ever seriously proposed was built, at its foundation, on variations of that design. The octopus breaks every assumption in the model. It has no centralized brain in the usual sense. It has no spinal cord. Its sensory processing is distributed across 9 ganglia. Its arms make decisions the central brain never approved. Its skin may see. Its genome reshuffles constantly. And despite all of this, it passes every behavioral test for sophisticated cognition that the scientific community has developed: tool use, learning, memory, planning, social recognition, personality, and possibly dreaming. If consciousness and intelligence can arise in a system this different from the vertebrate brain, then the architecture of the vertebrate brain is not a requirement for consciousness. It is one way, possibly a very narrow way of getting there. This changes how we should think about finding minds elsewhere. The search for intelligent life beyond Earth has traditionally focused on signals or structures that match what humans produce. We look for radio transmissions, for light patterns, for geometry that suggests intentional design. We are looking for something that thinks the way we do. The octopus suggests we might be wrong about what thinking looks like. Intelligence built on distributed processing, without a central command structure, without long-term memory stored in a single organ, without social learning, without symbolic language, would produce something that solves problems, adapts, survives, and possibly experiences the world in a way that is genuinely unrecognizable from the outside. We would swim right past it. The ocean contains more examples of that possibility than we have even begun to catalogue. Scientists estimate that about 80% of the ocean floor has never been directly observed. Of the roughly 300 known octopus species, the vast majority have been described only from dead specimens, preserved bodies hauled up in research nets, dried in museums, photographed once and filed. How they actually behave, how they hunt, how their arms process information, what their personalities look like, all of that is unknown for most species. For the ones that live below 1,000 feet, almost everything is unknown. Deep-sea octopuses from the Hadal zones, the ocean trenches that drop down 6 miles below the surface, have been filmed a handful of times. What is visible in those brief clips does not match the octopuses studied in shallow water. The bodies are different. The eye shapes are different. Some produce their own bioluminescence. Some appear to have no ink sac at all. Scientists working in deep ocean research have described finding cephalopods they could not classify even to genus animals, so far outside the known catalogue that the footage required new descriptions. Each of those unknown species is a separate evolutionary experiment. Evolution ran the octopus lineage in dozens of different ocean environments, simultaneously warm tropical shallows, cold polar depths, hydrothermal vent systems, mid-water columns with no seafloor to crawl on, and each environment selected for different solutions. The distributed nervous system we studied in shallow water octopuses is the architecture of those specific environments. The animals in radically different environments may have built their distributed intelligence differently. What does a mind look like when it evolved in total darkness, under crushing pressure, with no light input for its skin vision system to process, and no opportunity to camouflage against anything because nothing can see it anyway? Does the arm intelligence still dominate? Does the central brain expand to fill the role the arm's local nervous systems play in shallow water species? No one knows. The data does not exist. The octopus that has already been studied has forced biology to rewrite its rules about intelligence, consciousness, evolution, genetics, and the law. The ones that have not been studied yet are sitting in the dark at the bottom of the ocean, living their one-year lives, solving their own problems in their own way. The most alien mind on Earth may still be the one we have barely met.